A couple of quick announcements before we get started. Strange how few people are here relative to Friday. First one is that the chemistry seminar this Friday, Lauren Williams from Georgia Tech, Molecular Symbiosis, RNA, Protein, and the Origin of Biology. Should be really cool. That's Friday afternoon at 3:15. Unfortunately, I'll be in Nebraska. Lucky me. I think it was 6 degrees this morning in Lincoln. So, yeah, my choice. And that means that pre-Dr. Iverson will be giving the lecture on Friday. What I'm going to do as far as this week is concerned, I'm going to try and do online quizzes, clickers. So those of you who are planning on just being here for clickers can leave now. There are no clickers today because what would I be asking clicker questions on? And then at least for Wednesday and Friday, if you don't want to be here, that's your choice. There, however, will not be a recording of the lecture unless you ask one of your friends very nicely as far as Friday is concerned. So, first two announcements. Next announcement is that those of you who are thinking about registering for the Recombinant DNA Techniques Laboratory, which I think should be called the Mutant Viruses from Hell Lab, because that's what you're actually going to be doing in that lab. That, at least until now, has always been have a co-requisite of the Recombinant DNA Techniques lecture. That is not the case. And so if you would like to take the lab but can't fit the lecture into your schedule, just send me your student ID is fine, ID number or that Odin ID, and we'll make sure that that gets taken care of. So if there's some interest in that, yes, over there. Yeah, so the cell bio lab or the microbio lab, that you do need to have, unfortunately. As long as you can, unless you can prove, you know exactly what end of a micropipetter is which. Now maybe we can talk. Yeah. So the question is, sorry, just repeating it so it's on tape. Do I recommend taking the theory class as well? Yes, I highly recommend taking the theory class as well. The problem is it takes all quarter to do what's covered in the lecture in a week. So the actual timing of this. So that's the only reason. But yeah, no, the molecular techniques, we're going to spend like a lecture at the end of the term talking about this. But it's one of these other things. You could spend a year talking about some of this stuff, and it's moving really fast as well. Other questions about classes, et cetera? A couple of people have asked me about virology next term. Yes, I will be teaching it. Yes, it will be from 9 to 10:05 Monday, Wednesday, Friday, probably in a horrible room over in Kramer Hall. I'm working on changing that. But a number of people asked me about cell biology in terms of taking that at the same time, or if you have to actually have it beforehand. The answer is no, it certainly helps, but it's certainly not required, whereas I do require that you've had this course, but you're all here, so that's pretty straightforward. Okay, other questions about sort of registration, new courses, etc.? I do have your exams, or the Scantrons anyway. I'll be distributing them at the end at the back. So that brings us to canceling this thing here, because... Where are those lectures? Where is that going to be? Which? The Lauren Williams. Oh, Lauren Williams lecture? Yeah. That will be in room 107 in Science Building 1, so the lecture hall on the ground floor in Science Building 1. So I wish I would be here. He gives a great talk, and it's really cool stuff that they're doing in that lab. Okay, speaking of the midterm, as always, this is basically just the same thing. I've changed the numbers a little bit because that's where we are right now. The current high score is 90, but clearly that's a big outlier in terms of the two exams. So I'm just going to normalize to 80. You can do the math. 90% of that is 72, 85% is 68, etc. So you can get yourself an idea where you stand. Again, clickers are going to be about 10% at the end. I've told a number of people online I try not to change letter grades too much based on clickers. So yes, they're quote-unquote 10%. Any questions on sort of grading exam stuff? I have scanned all of the Scantrons, not just for scoring, but also have a image of them. So if there's any problem with the actual scoring on those scantrons, let me know, and I will go back and check them. Yeah?
So when you said normalizing to 80, does that mean that 80% equals an A, and you basically figure out percentages of 80%?
No. So I set 80 points to 100%. Oh. And then you do the calculation based on that. Yeah?
So for at least one of the keys, there was a question.
Yes, all three of the keys had one question X'd out. That question doesn't count, so the top possible score on the exam would have been 49. The high score turned out to be 46, so it doesn't really matter. Other exam questions. Okay, so see if I move forward on here. If it will actually let me do this. I'm going to keep trying to use this touchpad, and we'll see how well it ends up working or not. See? Not terribly well is the answer, I think. No. So for advancement anyway, we'll do this. So finally, hallelujah, we're talking about regulation, sort of what we've been leading up to all term. It's all about the DNA. Well, it's not quite true, but that's close anyway. And if we've got some time at the end, I'll show you this really geeky video that some friends of mine at Baylor College of Medicine made. But it's basically all about the DNA, or all about the base. So rest of today, we'll talk about differential regulation. And so the key really here is that, yeah, sure, all the cells have basically the same DNA to them, but what determines what cell is what cell, how that cell particularly responds at any given point in time, under what environmental conditions, really depends on the regulation. What pieces of those DNA are being turned into either active RNA or turned into that active protein. A lot of this has to do with transcriptional regulation. And so we'll talk a lot about, mostly the rest of today, DNA binding and how proteins actually interact with DNA. And it's not about taking the two strands apart, which is everything we've talked about so far. It's keeping those bases together and looking at binding, particularly in the major group, but some in the minor group as well. Talk a little bit about dimers and some DNA binding motifs, cooperativity, and if we get to it, some nucleosomes at the end. So the main thing here is really about this amazing diversity, and this is just the example that we have from human cells, but you could think about the same thing in terms of bacteria under two completely different environments. These guys have exactly the same DNA. The neuron has the same DNA in it as the liver cell does, but clearly they look really different relative to each other. They do have some of exactly the same proteins which are being expressed, and in fact these are most of the proteins that we've talked about so far. So these are going to be your RNA polymerases, your DNA polymerases, all of your general transcription factors, etc. And certainly from a basic metabolism point of view, you're going to have all of these proteins. You're going to need all the DNA repair proteins in all of these cells. But what makes the neuron different than the liver cell are all of those other proteins that are being expressed. Everything else which is in there and is leading to differentiation, responses, etc. So it's all about how each of those things get turned on or turned off. And there's been some really nice experiments. Most people think about Dolly in terms of cloning, but in fact, these were experiments that were done long before Dolly, basically showing that it really is just the DNA that you need. And if you take the DNA from one organism and put it into another cell, you end up with not the organism that was based on that cell, but the organism was based on the DNA that came into it in the first place. And so these are results from John Gurdon, who got the Nobel Prize, I think, a couple of years ago now, basically for this process. And all this really is, again, the same kind of process that we've talked about, hopefully everyone knows about in terms of Dolly, which we'll take a look at in just a second. But you take cells from an adult frog, and in this particular case, they did this from one species of frog, then take an unfertilized egg from a different species of frog, get rid of the nucleus, and this is sort of the big change between these experiments and then the somatic cell nuclear cloning experiments, which we'll talk about in just a second. And then you take this nucleus from these cells, put it into this egg, and yeah, there are a lot of extra steps here and magic occurs. And then you end up with a pretty normal embryo and a tadpole, but that tadpole has the genetic basis of this, sorry, not of these nuclei from the adult here, as opposed to what's coming in here from the egg, with one big difference. What is that? Mitochondria. The mitochondria, exactly. And so there's actually been some really interesting stuff. I don't know how many of you have seen this in the media about some kids with 3 parents. And they're in the process of setting up some of these in vitro fertilizations in England right now. So mitochondrial disease, mitochondria, of course, come all in the maternal line. And if the mom has some genetic problems in her mitochondria, then they're looking at, in fact, doing some experiments where they get the genetic material from the 2 parents, then put them into an egg which has mitochondria from a 3rd parent. So there could be some kids who literally have 3 biological parents, which is, and this is happening. This is stuff which is literally going on now. So plants, turns out, are way easier to work with as far as this is concerned. You can do very similar kinds of things. People haven't done this nuclear cloning process, but it turns out that plants are really, really easy to clone. And by clone, basically, so you take one cell, and this is the main reason here is you can get differentiation. So differentiation, let's back up here real quick. Here, differentiation is where you go from the egg to liver cells and neurons. And a lot of people thought once you went through differentiation, that was it. But clearly, these experiments show that at least the nucleus is still just fine. And here, individual cells, and this is particularly a carrot, but it turns out it can work for pretty much all plants. Again, always exceptions. This is biology here. But you can take a single cell, and that will eventually develop into a whole new plant. I don't like the whole carrot thing here at the end because this, of course, is just the root. But it will develop into a whole complete plant. So if you just take one cell from one part of the organism, it can develop into a complete plant. Turns out it's not true for all cells, but the vast majority of them. This is what people normally think about when you think about cloning. This is our friend up here. Quick trivia question. No, you don't need your clickers for it. Is Dolly the big one or the little one? The little one? How many votes for the little one? How many for the big one? Okay, the big one is correct. So this is basically showing that Dolly is normal because she can have babies, lambs, that are, as far as we can tell, actually completely normal. So this is also the picture that came from the Nature paper when they originally described this. But the basic idea here is just shown with cows, but this could just as easily be sheep. Cows is what they use in our textbook. You take cells from the oviduct, which again, they have to be actively replicating cells. Turns out you can't do this with neurons. Like neurons is really kind of a bad example to use at the beginning here. But these are clearly adult cells, and these adult cells you can remove. You can either take the nucleus out, or in this case, they just put the cells right next to now a completely enucleated egg. And so here what they literally do is they suck out the nucleus here. And so you have a completely enucleated egg. Then you put them together. You zap them. This is sort of the magic zap. And it's not completely clear exactly what this does, but if you don't zap them, you don't get this taking place. And now you end up with an egg with a nucleus in it, which has come from the adult. And this undergoes cell division, and you end up with a, as far as we can tell, pretty normal organism that you get back out of it. And again, the idea here is that Dolly is able to give birth to perfectly normal lambs. A lot of people discuss this. What about the epigenetics? What about the modifications? What about accelerated aging? Dolly lived to a ripe old age of, I think, 12, which for a sheep is pretty old, and ended up dying from an infectious disease that was spreading through the flock at the time. Yeah?
You say basically normal. What differences are there in the next?
So what differences are there in the offspring? And so the reason that I'm mentioning here basically normal, as well as Dolly, there was also Polly at the same time. Polly was then a genetically modified sheep at the same time. Here, Dolly, there was no genetic modification whatsoever. It was just the nucleus from one breed of sheep being put into the egg of a different breed of sheep. But her litter mates, if you want to call them that, were then genetically modified, because the whole point of this was actually to try and produce human therapeutics in the milk of the sheep. That was the whole point of this cloning exercise here, was to do this. Now, I say cloning, we'll talk about molecular cloning a lot more later on. This is what most people will talk about when they talk about cloning, but this is really somatic cell nuclear cloning. It's all about moving the nucleus from one organism into an egg of another organism. And so that's, again, SCNC, some people call this, but somatic cell nuclear cloning. We talk about molecular cloning, and most of the time when you talk about a molecular course and the recombinant DNA techniques lab / mutant viruses from hell lab, that is really about a molecular cloning and getting that exact DNA which you're adding to other pieces of DNA. And that's really where the recombinant aspect comes into this. So this, I think, is a pretty good indication that, yeah, it really is just the nucleus. And the other question, getting back to this one here about what were the differences, there clearly are some epigenetic markers on some of this DNA. The telomeres on an adult cell are somewhat shorter than the telomeres that you would normally have in an embryo, and people argue a lot about what this means. But as far as we can tell from most of these animals that have been generated by somatic cell nuclear cloning, they seem to be pretty normal. Okay, so if it is all about the DNA, what about that DNA is different between the different cells? Or if it's not the DNA, what is different? And, gee, surprise, surprise, it's mostly the RNA. And so this brings me to some new techniques that have been developed literally in the last couple of years, which are sequencing ridiculous amounts. When I say ridiculous, I mean millions of sequences of RNA, or usually it's the DNA copy of the RNA which is being sequenced here. So basically you get the RNA from any given cell at any given time, change it into DNA, and sequence the ... out of it. And what you find are lots and lots of sequences which correspond to sequences that you have in the genome. And so this is the example for beta-actin here. These bars down here, going from nothing up to a very large number, represent the number of sequences that were found that match the sequence of the DNA here. So they basically say this is RNA, the amount of RNA which is made that corresponds to this part of the genome. So here you see in the excuse me, this is exons, the red bits here, lots of RNA. Here, introns, much less RNA. And that's true in lots of different cell types: stem cells, liver cells, muscle cells, blood cells, etc. You don't see expression in the introns. You do see expression in the exons. One interesting thing, you see also a lot of RNA down here at this end. And we'll talk much more about this later on. Most of this has to do with translational regulation. So you've got your start of transcription here. The cap gets added. You've got quite a lot of RNA before you actually get to the protein coding sequences. And this, of course, is where that ribosome is normally scanning along after it binds to the cap and scanning along through here. So this is true for some genes. Again, this is beta-actin, and actin you need for cytoskeleton. So almost all cells have this. On the other hand, you have some genes, this is just an example thereof, tyrosine aminotransferase gene, which is only expressed in very few cell types. So here, liver cells, for instance, and here you see just sequences from these exons, no sequences from the introns, and in fact, you do have a little bit of expression over here at the end. But here, nice example of how we've got differential expression of one particular gene, and this is, again, by just looking at all the RNAs which are being made in each of these individual cells, and then looking how many times you find one particular sequence. And so it turns out in liver cells, you find the sequence that corresponds to this exon and about the same level of all the other exons of this particular gene. But you don't find it in lots of other ones. You can do this for every individual gene. It's kind of a pain. It takes a rather long period of time, although computationally we're getting much better at it. What more people do are these so-called microarrays. This is now looking again at the DNA copy which has been made from the RNA in any particular cell type. Turns out in this particular case, it's lots of different cancer cells. Each of the lines here represents one gene. So all the rows here, we've got, I'm sure you counted all of them, there are 1,800 rows. So here, all that you're looking at is the relative amount of that particular RNA relative to a background level. So black is the background level. It's because there's no difference between the expression in one particular cell, and this is, say, prostate versus lung versus brain, all these different cancers. Red is it's now expressing more of that particular gene. In fact, there are a bunch of different genes here, for instance, in the prostate, which are being expressed more than the mean, and quite a few genes that are being expressed considerably less than the mean. And one of the things that you can hopefully get out of this is that any given, say, cancer type, leukemia, it's a bunch of genes that are expressed similar to each other. Each column here is a different cell. Brain, similar kind of expression. Lung, similar kind of expression as well. And so you're just looking at the RNAs here. And RNAs, a great way to go. We'll talk about regulation in a couple of minutes. It's really a lot about the RNA, but the RNA doesn't do very much. In most cases, it'll be regulatory. It's really much more about the protein. And that you can look at on these gels. We actually had the thing that will work here. These are now looking at, this particular one is about 800 proteins that you're looking at simultaneously between two different cell types, liver versus brain. And basically each of these spots represents one protein or a slightly modified form of that protein. And we'll talk about how you make these kinds of gels towards the end of the course. It has to do with the isoelectric point and how big they are. All of the red spots are the same in both of these. Maybe a little hard to tell, but these red spots right here correspond to these red spots over here. So these are proteins which are expressed both in brain and in liver, but then you've also got a number which are expressed, say, just in liver. Here's some of those genes over here, proteins I should say, which are completely lacking over here. And this whole stretch of different dots here probably is some kind of modification. You think about an isoelectric point that has to do with the charge of your protein. These will be different numbers of phosphorylations, for instance. You see all these different things. The problem is it's a lot harder to look at these kinds of things than it is to look at the microarrays here. It's a lot easier to separate each of those individual genes relative to each other. So this is probably a higher resolution way of looking at what proteins are being expressed, and certainly proteins are doing the job for most of these things. It's a lot harder to study the proteins than it is to study the RNAs. So usually we're going to study the RNAs. There's another reason to study the RNAs, and that's because that's where most of the regulation is going on. And that makes sense because this is before you go through all of these extra processes. So once you transcribe a gene, you have a transcript, that gets processed, cap spliced and tailed, that gets exported, that then gets translated, the activity is being regulated, sometimes the amount of the RNA is being regulated, and then the final protein here is often modified as well. So starting way back up here is great in terms of conserving the energy. On the other hand, controlling activity way back here, your gene expression, takes kind of a long time because you have to transcribe the gene, you've got to splice the gene, you've got to get it out of the nucleus, you've got to translate it, etc. So here, this kind of control is much, much faster. If you want to have a very quick change in terms of what's going on inside the cell, you're going to want to control the level of protein activity. If, however, you care about conserving your energy and doing something like the different shapes that you have in a neuron versus a liver cell, that's something which is going to stay pretty stable for a long period of time. So you're going to want to control those guys here at the level of transcription, whereas the response to a neurotransmitter you might want to do in terms of changing activities of protein, although it turns out that's way too slow as well. These are electrical changes which happen. So we're going to mostly concentrate on transcriptional regulation, A, because it's a lot easier to study RNAs than it is to study proteins, and B, because it seems to be really this critical first step which is going on there. So how do you control transcription? Well, transcription, of course, is DNA going to RNA. What do we use? We use transcription factors. And this is, again, a horrible name, but again, it's what's in the literature. It's what's been used for years. These are now the specific transcription factors. We talked about general transcription factors before, the TF2, A, B, D, E, F, H, etc. So those are your general transcription factors. These are the specific transcription factors. So also known as gene regulatory proteins, about 10% of most genomes are dedicated to, at least the protein coding part of most genomes, are dedicated to these regulatory proteins. So there are huge numbers of them. One of the things that makes actually working with them really kind of hard is it turns out that there are very small amounts of them. So you've got large percentages of genes, but very small amounts of any of these given proteins, and it makes actually really kind of a pain to work with them. These gene regulatory proteins bind to normal double-stranded DNA, and most of the binding happens here in the major groove. I didn't bring my model over, but that model's got so many problems that probably decided it wasn't so useful. In the major groove of DNA, that's where you have all of the chemical information which you need to determine what base pairs are there. And we'll take a look at that in just a second. A lot of proteins will still bind a little bit to the minor groove, but those are going to be much less specific kinds of interactions. These binding sites, also known as the sequences, are located right next to the genes that they're going to be regulating. And next to is a kind of flexible definition. They can actually be thousands of base pairs away, but they're next to the genes that they're regulating. They're not completely distant in distant parts of the genome for 90+% of them. These gene regulatory proteins are usually extremely specific. They will bind to one sequence or a family of very similar sequences and then regulate the genes that are next to them through that process. This is, remember correctly, a slide which I showed before. Basically, again, looking at all of our base pairs. This is why I wanted you to learn the base pairs for the first exam. Because here in the major groove of DNA, this is just GC base pair AT, CG, and TA, you have all of these atoms which present a surface here which is different for every single base pair. So even a GC base pair is different from a CG base pair because of where these hydrogens are, which could be forming hydrogen bonds. We've got the, hang on, this should be donor versus acceptor. I mixed this up. I did this last year and I forgot to edit the slide. Silly me. Okay, this is blue is the donor, of course, because that's the hydrogen atom which could be donated into a hydrogen bond, and red are the acceptors. So these guys could be accepting hydrogen bonds, these guys could be accepting hydrogen bonds, and clearly here it already is. But if you just look across from one side of the major group to the other one, we've got acceptor, acceptor, donor, and here, the opposite one, you've got donor, acceptor, acceptor. So these two look different to something coming into the major groove, and of course that something is usually going to be a protein. Same thing is true for AT and TA base pairs, except now you have hydrogen bond, acceptor, donor, acceptor, and then this big hydrophobic methyl group sticking out in the major group. And in fact, if anyone were to give you a DNA model and ask you where is the major groove, one of the easiest ways to find it is just to look for where that methyl group is, because the only place that you find methyl groups in DNA is going to be sticking out in the major groove. So really good way to find the major versus the minor groove here. What interacts with these? Again, I mentioned this already. These are the side chains of proteins which interact with them. Asparagine and arginine are two of these that happen really quite often. This is not a typo, the capital N here, because that's the single letter code for asparagine. And the arginine, which we all remember, of course, is R. Asparagine has a hydrogen bond donor and acceptor, and arginine has two hydrogen bond donors. And that's just what you can see up here. Here's an example of a protein interacting in the major groove with a TA base pair. It doesn't have to be a TA base pair that the asparagine is associating with. It's just something that's going to have a donor and acceptor the appropriate distance apart to interact with this asparagine side chain. Same thing is true here for arginine, again, interacting in the major groove. This is all weak interactions. There's no covalent interaction here that happens between DNA and these regulatory proteins. It's always going to be non-covalent interactions, which means these are the weak interactions. In order to get enough binding energy, you have to have lots of weak interactions. So it's on the order of 20 or 30 weak interactions which are going on there. And because protein amino acid side chains can rotate back and forth relative to each other, hydrogen bond donors and acceptors, there are lots of different amino acids that can do this. It doesn't always have to be to the same base pairs. It could be two base pairs right next to each other. Turns out it's really hard to predict what amino acid sequence is going to bind to what DNA sequence. It would be so wonderful if you could do that, but it's unfortunately not the case. People are getting much better at designing proteins that can bind to specific DNA sequences, but it's still kind of hit or miss in many different ways. And an example of one of the ones that we know about, but this is only because people have studied an already existing DNA-protein interaction, is this example right here. We already looked at this asparagine interacting with the AT base pair. Again, hydrogen bond donor here from the base, hydrogen bond acceptor here on the base. But in the same process, this is our asparagine right here. It's about a 60 amino acid protein that's interacting with lots of different bases. Here, lysine, tyrosine, arginine, all of them interacting with different bases here. And we've got some interactions in the minor groove. This is supposed to be representing the other side of the DNA here, where you've got different amino acids that are associating here. Some of these interact with the bases. These are the arrows towards the bases. Some of them interact with the sugars. Some of them interact with the phosphates. So it's really, again, difficult to predict how these things are going to interact with each other. That being said, these interactions are some of the strongest non-covalent interactions that we know about, these two interactions that happen with each other. There's the antibody-antigen interactions, which in fact are really similar to this, again, non-covalent interactions, but DNA protein and DNA binding proteins are also very, very high affinity in terms of their interactions with each other. That's part of the reason why people are really interested to try and figure out how these are actually interacting with each other. Turns out that most of these interactions are not terribly surprisingly, and this is probably having to do with evolution, is that turns out that the width of the major groove of DNA is almost exactly right to have an alpha helix stuck into it. Where are the amino acid side chains in an alpha helix? They're sticking out on the outside. So if you've got amino acids sticking out on the outside and your alpha helix sitting in the major groove, it's a great way to have amino acid side chains interacting with the base pairs that are present in the major groove. Turns out that there are lots of different ways then to get an alpha helix into the major groove. We'll talk about a couple of them here. These are the most common, and again, these are things that have been found in DNA binding proteins and mostly DNA regulatory proteins here. The most common one you find in bacteria is the so-called helix-turn-helix. So it is just like it sounds. We've got two helices, alpha helices, N-terminus to C-terminus. The red one here is what's called the recognition helix because it's the one that really sits down right in the major groove of DNA and then has this other helix basically sitting down on top of it, holding it into the major groove of DNA. Probably about 90% of bacterial gene regulatory proteins have this helix-turn-helix motif. And this brings us to a discussion of what's a motif. A motif is a stretch of sequence which has a function but actually doesn't have a stable structure to it. And so there'll be parts of a domain. That domain has the full structure. You'll have these motifs that are present in there. Often these will be sequences as well, and you can see the sequences and recognize what those sequences are. Quite often you can actually predict that this amino acid sequence will form two helices, and you've got a helix-turn-helix, and you could predict that this would be binding to DNA, but you don't know what sequence that particular one's going to be binding to. And this, again, is a very common thing that you find in bacterial and bacterial virus DNA binding proteins. Here again, your red helix is the one that binds to the DNA, and then a blue helix that sits down on top of it. So here's a couple of bacterial virus proteins. Here's a gene regulatory protein. Here's another gene regulatory protein. Almost all of these are going to bind as dimers, which is getting ahead of myself here. We've got two polypeptide chains, which are identical to each other, that now interact. And we talked about dimers, or hopefully Eric Iverson talked about dimers before when we talked about proteins. Two, in the case of identical proteins coming together, that's going to be a homodimer. The different proteins coming together are going to be a heterodimer. These bind to each other and then bind to DNA, and surprise, surprise, they're almost always binding to parts of DNA which are about 3.4 nanometers apart. Why? It's because it's one full turn of the DNA. It's 10 base pairs between here and here. And so this alpha helix here is going to have side chains that interact with the bases in this major groove. This alpha helix is going to have side chains that interact with sequences over here in the major groove. Why have dimers? Well, the main reason for having dimers is that each of these alpha helices sitting down in the major groove binds to 4 to 5 base pairs. Well, 4 to 5 base pairs is great, but if you have a genome like E. coli's that has 5 million bases in it, you're going to find sequences with 4 to 5 nucleotides at random all the time, all over the genome. So in order to have your gene regulatory protein bind just to where you want it to bind. Now you have your sequence 4 to 5 nucleotides right next to another sequence with exactly the same sequence of those 4 to 5 nucleotides, which is now 8 to 10 nucleotides, and that you're only going to find 1 or 2 times in any particular genome. So here is a nice example of a dimer. This particular one is the control of repressor and other genes, just a bacterial viral protein, in fact, one of the first ones to be well studied. Binds to DNA as a dimer. It binds to 2 what are called half sites. So this is AT, AT, CG, AT, CG. This now binds to exactly the same sequence, but now on the opposite strand, over here, 5' AACAC. And again, it's the same thing as you have 5' AACAC. These inverted repeat structures means that this part of your Cro protein, again, this is going to be that alpha helix, the recognition helix, sitting down in the major groove of DNA, that's going to bind here and have identical binding to these sequences over here, but in the opposite orientation. And if you go back and look at these dimers, you'll notice almost all of them are mirror images of each other. So you see this one folded over like that, this one folded like this, etc. So they're all mirror image of each other. They're all interacting between these 2 half sites. Another thing to look at here, this sequence, 5' CGT, is now not the same as this sequence 5' GCA here. So that tells you that we have a protein-protein interaction. That's what's supposed to be here on the back side. And then a protein-DNA interaction. So this is how you can get your specificity. Yeah?
You just answered my question.
Okay. So that's true for bacteria. Not surprisingly, the eukaryotes are lazy. Well, probably came after the bacteria as well, though we can discuss that at length some other time. Basically, the eukaryotic equivalent of this helix turn helix is what's called the homeodomain. Now, this tells you something immediately. It's a domain, which means what? Okay, it has a certain function, but some of those motifs have certain functions as well. But what about a domain? What's different between a domain and a motif?
Conserved sequence.
Okay, the conserved sequence is true in motifs and in domains. Has a stable structure. So that domain has a structure. So if you just look at a helix turn helix by itself is not going to be a stable structure. On the other hand, a homeodomain, which is 3 helices, actually is a stable structure. And you have a number of proteins which are just this homeodomain, so a single domain protein. And we looked at those way back when we looked at conservation of structures, like that 60 amino acid protein I talked about with the interactions in the major and the minor groove. That was actually this one right here. So here, recognition helix, sitting down in the major groove, interacting as usual with base pairs in the major groove. Here we have interactions now in the minor groove of part of that homeodomain protein as well. But it's basically alpha helix sitting down in the major groove. These guys can form dimers as well, but they can also bind as monomers. But again, the specificity is not going to be great because they're only going to bind to 5 or 6 bases. That's all that you can get here. If you just look at the major groove, here are their base pairs. 1, 2, 3, 4, 5. Any more than that, you're not going to be able to interact with. So probably the most common, again, of these proteins that are binding into the major groove of DNA, certainly in the case of bacteria, are either 2 helices in terms of the helix turn helix or 3 helices that you have with your homeodomains. On the other hand, there are a number of other alpha helix in major groove binding motifs. Probably the next most common of them is the so-called zinc finger. And I remember, well, we'll get back to this in just a second. best question I ever had from my undergraduate biology course, right, a few years ago now. But I still think it's a good question. So these zinc fingers make alpha helices, and these alpha helices sit down in the major groove of DNA. Why are they called zinc fingers? Because they all have to have zinc atoms associated with them. These zinc atoms bind to very well conserved sequences. And these are amino acid sequences. They're either going to be 2 cysteines and 2 histidines, or 4 cysteines. And so just back up here a little bit. This is cysteine, any amino acid, cysteine, histidine, any amino acid, histidine. And these are actually really great for a couple of reasons. One, cysteines are actually pretty rare if you look at protein sequences. Histidines are rare too, but not as rare as cysteines. Even rarer is that you find them that are literally 4 amino acids apart. And so if you have a protein that's got a sequence motif in it that looks like this or looks like this, it's probably going to be a zinc finger and probably going to be causing something to bind to DNA. So just by looking at the sequences, you can make a prediction that this motif is likely to be binding to DNA. There are 2 different kinds of zinc fingers. There are those that have 2 beta strands and an alpha helix, where this alpha helix binds to the DNA. You also have some that have 2 alpha helices, and then each of those alpha helices bind to DNA. We're going to talk a lot more about this particular protein right here. It's called the glucocorticoid receptor, in terms of other interactions with DNA and how that's being regulated a little bit later on. But it's a number of different transcriptional regulators that are there. There are a couple of other ways you can put alpha helices in the major groove of DNA. One of these is here. It's the so-called basic helix with a leucine zipper. We already talked about leucine zippers. These are those alpha helices that every 7 amino acids have a large hydrophobic amino acid residue that then interdigitates. And so these are your leucine zippers, and this is where I get to mention the question, I think it was actually on the final exam. Can a zinc finger get caught in a leucine zipper? Geeks, all of us. So here is the leucine zipper interaction, but this is just involved in making a dimer. Has nothing to do with binding DNA, and a lot of people get confused by this. The leucine zipper is a DNA binding motif. No, it's a dimerization motif. In many cases, it also has, and often even in the same alpha helix, a basic domain, which is right here. And basic domains, what do those have? Basic is going to be what? What kind of charge is something with basic amino acids going to have? Positive charge. What does DNA have? Negative charge. And so these are basic amino acids in this alpha helix. Now, just charge interaction is not going to give you any specificity, right? So it's going to have some charged amino acids to help it bind to DNA, and particularly the backbone here. But then it'll have other, usually going to be hydrogen bond donor and acceptors, which are present here in the major groove. They'll be interacting with the base pairs. So we've got these 2. Also, you'll notice this is a dimer, so we're going to be able to interact with multiple base pairs, and also in an inverted orientation to each other. So if this sequence is binding here, that same helix over here is going to be binding that sequence there, also inverted repeat. Another way that you can end up with dimerization is this so-called helix-loop helix. Again, dimerization motif, helix-loop helix. These guys bind together as a dimer, and very often they'll have a basic helix which is right next to them. And again, this basic helix is going to be interacting with DNA. So leucine zippers, dimerization, helix-loop helix, not to be confused with helix-turn helix. Sorry about that. But the helix-loop helix is dimerization. Helix-turn helix is DNA binding. And it has to do with the differences here. So this is, in the turn, you just have a couple of amino acids between the two helices. And here, it's about 10 amino acids that's present in this loop structure. Dimerization is really great for specificity because you can have your two half sites sitting right next to each other, get much higher specificity. You can have the same protein that is now going to be able to bind to 5 nucleotides or 5 base pairs. Put it together, you've got 2 that bind to 10. But even cooler about these dimers is that you can mix and match them. So it's what's called heterodimerization. So we talked about homodimerization. There's 2 of the same proteins interacting with each other. They're going to be binding to the same half sites. If you now have dimerization motifs that can interact with multiple different proteins, and I think the easiest way to think about this is, again, with leucine zippers. So you've got a stretch of amino acids. Every 7, you've got a leucine hanging out on the side. But the basic helix that's right next to that may or may not have the same sequence. So you can get dimerization that can happen either as a homodimer, which we have down here, the dark greens, or a different homodimer. Here in light greens, these bind to blue half sites and red half sites relative to each other. Or you can do a mix and match, where you've got the dark green and the light green, which are now going to be binding to blue and red right next to each other. And if you think about multiple different proteins, each of which has a leucine zipper that can associate with it, you can clearly have many fewer genes, but mixing and matching all of these can give you many different binding sites that you can in fact interact with. And we'll talk about many more of these later on. Probably the best known of these was something called phosgene, which is a oncogenic protein that's very important in a lot of cancer development. It doesn't have to be these leucine zipper interactions with each other. We'll also talk about the MAT alpha 1, MAT alpha 2. This has to do with the control of yeast mating type. Who cares about yeast mating? But we'll get back to that. These are 2 very different DNA binding domains that are associated with each other, again, through heterodimerization. Yeah?
Is the leucine zipper the entire dimer, or is it just the binding site where the dimerization can happen?
So the question here is basically, where is the leucine zipper relative to this? So the leucine zipper is what's up here. It's that dimerization interface up at the top here. So it's where the 2 alpha helices are wrapping around each other. Every 7 amino acids, you've got one of these large hydrophobics, usually leucine, but not always leucine. You can even have some examples of a heterodimer that probably in evolution has ended up being attached to itself. So this is one particular protein here that actually has 2 different DNA binding domains. And again, domains, they can have separate structures, stable by themselves, but they're connected with an amino acid side chain so they can interact with each other. So you've got these heterodimers in one individual protein. So what do these guys bind to? Again, we talked about half sites already. It's an example now again of heterodimerization down here. Here's one dimer, homodimer interacting with this site, homodimer interacting here, heterodimer interacting here. This is a consensus sequence, which again we've talked about for promoters already, for a half site of one particular gene regulatory protein. This one's called NANOG. Turns out that expression of NANOG, and I think the latest count, there were 3 more gene regulatory proteins. If you take a differentiated cell and express these 4 proteins, including NANOG, they can become pluripotent stem cells. It's really amazing. Expression of 4 gene regulatory proteins can lead to de-differentiation and making stem cells. Absolutely amazing stuff. Susumu Tonegawa got the Nobel Prize for this about 5 years ago now. So NANOG is one of those proteins, and it binds to this consensus sequence, which in most of the cases it's TAA, TTGC, but it can also bind to CAA, GG, GG, etc. always in positions 2 and 3 it's AA, almost always at position 1 it's T, etc. And this can either be now something which will be a homodimer, and again this would be 5' TAA TTGC here, and then on the opposite strand TAA TTGC there, or you could have a different sequence over here that's going to bind to a different half site. If you look at dimers binding to DNA, just looking at the amount of binding that you have relative to the protein concentration, if you have normal binding, again, this could be a single protein or very stable dimers binding to DNA, as you increase the protein concentration, you slowly increase the amount of binding here. But we've talked about a little bit about protein cooperativity before. We talked about the single-stranded binding protein. Basically, once you bind one protein to the DNA, you get a whole bunch more proteins which are going to bind to it as well really quickly. Same thing is true for nucleosomes as well. Also is true for a lot of DNA binding proteins as well, these gene regulatory proteins. And so what happens here is you have an equilibrium between monomers and dimers, but as soon as you bind to DNA, then you have a vast increase in this amount of dimers here. So the dimerization is stimulated by DNA binding. And what this does is basically, instead of making a sort of slow progression from low concentration to high concentration, you'll have the regions here, you've got partial binding. This makes it much more of an on or off switch. At low protein concentrations, you have no binding because these guys can't find each other. At high protein concentrations, you've got tons of dimer around, so you're going to get lots of binding. And you go from this no binding to all binding in a pretty narrow range of concentration. And usually, you don't want to have some genes that are turned on a little bit some of the time. You're going to want a gene which is turned on or is turned off. And this is just a molecular way of doing that, is it's through this cooperativity and giving you much more of an on-off kind of process. The last things I wanted to talk about today are interesting things that I don't have any images for. Okay, good. Well, this is basically about the same thing here. So the problem that we have, and of course all eukaryotes, is that our DNA is complexed in nucleosomes. Also, bacteria have chromatin, things which are being compressed as well. And basically, you have to have these DNA binding proteins, the gene regulatory proteins, that have to be able to interact with DNA which is bound up with nucleosomes. So there are a couple of ways that that can happen. This is the last one of those here, and the other pictures, which I'm not quite sure why they didn't transfer here, but has to do with nucleosome breathing, which we talked about way back when we talked about nucleosomes, and that is that the DNA comes on and off the nucleosomes, particularly towards the ends of the nucleosomes, with a certain frequency. It's a relatively low frequency, but comes on and off. And basically, if you have a DNA sequence next to a nucleosome, or it's bound to a nucleosome, if it's bound to a nucleosome but near the end, it's about 20 times less likely to bind than if it's naked DNA, i.e. not bound to nucleosomes. But if it's in the middle of that nucleosome, again wrapped all the way around, that's about 200 times less likely to be interacting there. One way to deal with that is to have other proteins which will bind here at the end where you have your breathing taking place. So here, the green binding site here. This now will bind when you have that little bit of breathing that takes place. But the binding of this protein, which now binds with higher affinity than the histones do to the DNA, will basically pull this piece of DNA off so that you can get binding of this second protein. And so this brings us to what we'll be talking about a lot more for the rest of regulation, is going to be the interactions of multiple DNA binding proteins interacting with these nucleosome-bound templates. And so with that, I will finish here and click over to this. I don't know if anyone's had a chance to listen to this yet, but let's see if we can actually get it going here. I think this is absolutely hilarious, but again, I'm kind of geeky here. I will also, once it actually loads here, we can, I'll distribute the midterms to the back, and you can listen to this as you like.
Because you know I'm all about that base, no proteins. I'm all about that base, no membranes. I'm all about that base, no lipids. I'm all about that base. Yeah, it's pretty clear. I work in base 3. The third can wobble, but still it's clear to me. Cause I got that charge that all the proteins chase. In all the right grooves, in all the right places. I see the helicase working its binding side. We know one S&P, it won't be folding right. If you got function, just let them see. Cause every ancient is dynamic from the 5' to the 3. Yeah, my structure is folding in helix with twisting right. It's the best way to keep the whole genome wound up inside. You know I won't be no stick model Watson Crick before. So if that's what you're into, then boy, you've got lots to learn. Because you know I'm all about that base, no proteins. I'm all about that base, no membranes. I'm all about that base, no lipids. I'm all about that base. Hey, I'm bringing Franken back. Go ahead and tell those Nobel winners that. No, I'm just playing. I know you think they're right, but I'm here to tell you, every angstrom is dynamic from the 5' to the 3. Yeah, my structure is folding in helix with twisting right. It's the best way to keep the whole genome wound up inside. You know I won't be no sick model Watson clicking before. So if that's what you're into, then boy, you've got lots to learn. Because you know I'm all about that base, no protein.
Well, don't we need proteins for base synthesis?
Not to mention DNA replication, repair, and transcribing RNA. You know, lipids and membranes do a good job protecting DNA.
Well, technically, DNA has ribose, which is a sugar.
That's a good point. Sugar is necessary and tasty.
Except, of course, genomics, epigenomics, and transcriptomics. Except for when ER has ribosomes.
Yeah, but smooth ER doesn't have ribosomes or bases. So it's all about the base. What about cancer?
Base mutations.
Nutrition?
ATP.
Cryptography?
The genetic code.
True love?
Complementary pairing.
Nuclear war?
The H-bombs.
What about Neil deGrasse Tyson? He's a physicist.
Have you heard his voice? The man's clearly a base.
I know, I want to learn it off too.
Yeah, so basically, 11 would also work.